First of all, thank you very much, Peter, for that very warm and I would say, to be a bit critical, over-generous introduction, but of course very enjoyable as always. It's a great pleasure to come back again to this wonderful meeting and of course meet many new people, but it is always also a pleasure to meet so many old friends here again, and I'm particularly happy to see here Professor Siert Klöting, former president of Academia Europaea and the greatest president we have had for Academia Europaea, to meet again Eva Conterosi, who is of course one of the people who also have contributed so much to making Szeged a famous place. And I'm also very happy to see Balázs Gulyás here. He has, on the other hand, kept me a little bit busy this year by evaluating a lot of institutes for the Hungarian Research Network, but that also has been a very interesting task. So come back to this a great meeting, and I think we cannot be enough grateful to Peter Hegyi and to Andras Varro for having masterminded this program. Sultan and I were talking over lunch. There was nothing, of course, like that when we were students. Nothing at all. So, I mean, you have already, all of you younger generation who are sitting here in this hall, you already have a big advantage compared to us old folks, actually, because you got a much easier start in life than we had. We had nothing like this program ever. And in fact, I mean, you have a particular advantage actually being in Hungary, because this program that Peter and Andras have generated is actually unique to Hungary. And this year, of course, it's a particular pleasure that it has been internationalized via the connection with Academia Europaea, yet another important achievement. Now, this slide here I want to use in a way to highlight the importance of having outstanding people in a particular place. It plays a huge role in the image we have our place. So actually, I knew about Szeged very early on, and now you will all think that I knew about Szeged because of Albert Szent-Györgyi. But that's actually not the case. I knew about Szeged because of the person at the bottom right of this picture, which probably most of you have never heard about it, and if you have heard about it, you might even have forgotten about it. But the person that I, as a teenager, knew, because I was a very avid interest in classical music, was Ferenc Fricsay, or Fricsay Ferenc, as you would say it. He grew up here in Szeged, conducted at this wonderful small opera theater you have here. And if you go to that opera theater, you will see the plaque that is here in the middle of this picture, commemorating that Ferenc Fricsay conducted here in this opera house. He went on to become a world-beating conductor and sadly died very early in his life, only 48 years old. And if he had lived longer, he would undoubtedly have been commemorated as one of the greatest conductors ever to be mentioned, in the same way as Wilhelm Furtwängler and Bruno Walter and Arturo Toscanini. I remember personally several unforgettable concerts in Copenhagen when he came and conducted, and these are still some of my most precious memories. In fact, when the city of Szeged were not interested in supporting the opera sufficiently, and that's a very nice story, actually, in terms of thinking about how art and science sometimes work together. Then Albert Szent-Györgyi, who was also a very cultural person, he, together with Fricsay, went to the city, complaining that it was a kind of cultural vandalism not to support the culture in Szeged sufficiently. And at least something was achieved. Eventually, unfortunately, Fritzsche left for Budapest and then left for Salzburg and Vienna and Berlin and Munich, and was in that sense sadly lost to Hungary. But he became the greatest interpreter of the very great Hungarian composer Béla Bartók. So anyway, my story is that these people, whether it was Albertson Georgi in science, Bartók, Ferenc Fritzsche, they sold, so to speak, Szeged and Hungary to the world. Because of people like that, and there are many, many others, of course, we have the perception of Hungary as a clever country, as an intelligent country, as a country that has contributed to civilization. And it plays, I think, a big role in the way a country plays on the world stage. So in this talk here, I will focus, because I cannot talk generally about everything in a confinement of a lecture. So I will focus on some molecules, transporters across the plasma membrane of cells. And basically we have three types of transporters. We have ion channels, we have active transporters, we have transporters that work by simply shuffling things along gradients. And for some reason, when I was a clinical medical student at the University of Copenhagen, I had somehow the inclination, I cannot describe it in any other way, that I would like to do something. This may not sound very impressive, but I wanted to do something a bit more than just do my studies. And so, through the somewhat chaotic way the Institute of Physiology in Copenhagen was governed on that time, which actually turned out to be a big advantage for me personally, it was possible actually, together with a friend of the same year as me, to conquer a laboratory. We did not ask permission of anyone. We went and took this room, which was empty. We went around the institute, collected some equipment, and we had some interest in electrophysiology. And we wanted to just do something that had not been done before. And for some reason, we eventually fastened on the salivary glands, a seemingly unimportant object, and also on a seemingly not very significant problem, but one that somehow interested us. We wanted to understand how salivary secretion is switched on and switched off. And it turned out actually that there was not much literature about it, which was very advantageous. There were only a few papers, which we read in one afternoon. And then we decided to go after this phenomenon. So it's a very impressive phenomenon when you do a simple experiment. You can isolate the salivary gland, you can isolate the nerves to the gland, you stimulate them electrically, and saliva pours out of the salivary gland. You stop the electrical stimulation, and the secretion stops. So we asked ourselves the question, what is actually happening inside the cell? It turned out that there was a neurophysiologist, a well-known neurophysiologist in Scandinavia, Anders Lundberg, who was the only one who actually had thought about this problem. And he had arrived at the conclusion, I don't think this pointer is working actually, but don't worry about it. So anyway, in the green box there, you saw his very simple theory. So he said there is a chloride pump sitting on the basal side of the membrane, and when the neurotransmitter acetylcholine released from nerve endings hits the membrane, it activates this chloride pump. So that was something that was in the time, it was also in the frog skin, it was in the cornea, that active chloride pump stimulated by some agonist would actually switch on secretion. And now comes, I think, what is potentially an important point. My tutor in physiology, who was a very critical person, but also a very interesting individual, he used always to go around and say, well, I permit myself, he said, to doubt the truth of what you say. And I think that's very important for science. So we sort of told him about this. He worked on something completely different. He says, I don't believe this here, and you should test this. Probably he is wrong. We said it was published in Nature, there was a big review in physiological reviews and so on. No, we said, I think it is wrong. Try to demonstrate that this is nonsense. Okay, we took up the challenge. And indeed, he was right. It was completely wrong. So we did a simple experiment, which actually had not been done. We just changed the external solution perfusing the salivary gland from one containing chloride to one containing sulfate. And we expected confidently, because we actually at that point thought that Anders Lundberg was right. So we expected the hyperpolarization to disappear. But as you can see in the sulfate solution, if anything, it became bigger. So it did not seem to fit very well. It was obviously not a final proof against it. That came only later with more work we had to do. But it was the first and clear sign that probably our friend and critique, Christian Krone in Institute, was right. It was plain wrong, actually. And that's, I think, the important lesson. Question things, even if they have been presented with all the authority of having been published in a top journal and so on. Ask questions. Is it really true? Now, this led, amazingly, in spite of the fact that this was kind of work done by two students who had no status whatsoever. And there were many people in the institute in Copenhagen who said, these two student professors, what do they think they are? They have disproved what a famous professor of neuroscience who worked actually with the Nobel Laureate John Eccles and had therefore enormous authority in a group. How can they think that they have actually been able to prove him wrong? So anyway, one day I received a letter from the University of Pennsylvania. It was the first letter I ever received of any official nature, inviting me to give a talk at the University of Pennsylvania in 1968. So that was my very first invited lecture, and I could present these data at that conference. And that led a little bit later to a second important invitation that shows again that one thing very often in science leads to another one. And I was invited to the Royal Society. It was my first professional visit to the UK. And this visit became very important because up to then, I actually had not found anybody who was working on a similar problem to the one I worked on. So that sort of gave the impression that maybe it wasn't very important. Nobody else was actually interested in it. I was lucky in the sense that nobody bothered about looking at the number of citations at that point, because the number of citations to my work was extraordinarily low and remained low for many years because it was not a big area of research, actually. But nobody cared about citation numbers at that point, and that was, in a way, not a bad thing. But at this meeting at the Royal Society, I, for the first time, met somebody who actually thought about the same kind of problems as I thought about it. And this was Mike Berridge, who later became very famous for his discovery of an important intracellular messenger, namely inositol trisphosphate, the ubiquitous messenger that releases calcium from cells. And so this meeting for the first time with Mike Berridge, and it became the first of very many meetings we met over the more than 50 years everywhere in the world and spoke at meetings together. And sadly, Mike died early in 2020, but I have had the great honor of writing his biographical memoir for the Royal Society, where I have honored his memory. He was a truly great investigator from whom also I learned so very much. Now, at that meeting of the Royal Society, actually, neither Mike Berridge or I had any final coherent theory of how salivary secretion worked. Mike worked on an even more esoteric gland than I did, because at least I worked on a salivary gland in a mammal, but Mike worked on salivary glands in the insect, and so one could hardly think of anything less important than how a salivary gland works in the insect. But it eventually led to a very big discovery. Early on in Copenhagen, I was given the opportunity to organize a meeting at the Royal Danish Academy, and I invited Mike to present his work there, and that was actually, in many ways, one of his first very great successes. First of all, the father of the concept of stimulus secretion coupling, Bill Douglas, a Scotsman who worked at Yale University for very many years, was there. And you can see his remark after Mike Berridge's presentation. which obviously in itself was quite important. So what Mike had focused on, and that became very important in epithelial transport physiology, was actually the discovery of the calcium-activated chloride channel in the apical secretory part of the membrane. And that turned out to be a very important discovery, although even that was forgotten later on, and this channel was discovered by other people working on oocytes and by neuroscientists who had forgotten that actually it had been discovered by Mike Barish many, many years before they did it. It took very many years before finally I could prove that this chloride channel exists specifically in one membrane, namely the apical secretory membrane. So this is already a big jump to many, many years later. We're still working on this problem and wanting to show in a very, very clear-cut manner that there was this calcium-activated channel and it was sitting in an exact place where it needs to be in order to play a role in secretion. And in this particular experiment, we, of course, technology had advanced enormously at that point, and we could put a calcium sensor inside the cell, we could put caged calcium inside the cell, we could uncage calcium, that is, liberate calcium from its binding state in a very specific subcellular position, and we could measure the overall chloride current. And as you can see in the right side, there's only one place where calcium uncaging actually switches on the chloride current, where the green arrow points. So this is a very clear and straightforward demonstration of what Mike had originally suggested many years before. But not to get ahead of ourselves, electrophysiology changed in a very radical way, and Peter already introduced this with the patch clamp technique. Up to the point when Erwin Neher and Bert Sackmann invented, one has to say, the patch clamp technique, all of us had been working with sharp glass microelectrode, which we pierced the cell membrane, and the electrode was inside the cell, and we could measure the electrical potential difference between the cell interior and the outside, or if we voltage clamped it, we could measure the current. And Erwin and Bert turned this completely around, and they approached it from the outside and put a patch pipette on the surface of the membrane, as you can see in the upper left part. So you basically electrically isolate a bit of membrane, and then with suitable amplification, you can actually be lucky enough to isolate an area of membrane where there are only 1 or 2 ion channels. And you can then, with suitable amplification, actually measure these tiny, tiny currents, picoampere currents, that pass through this bit of membrane. And at the lower left, you can see an early recording published in Nature in 1976. It was the very first single-channel current recordings from a biological membrane. And they already showed the characteristics. This channel is either in the open state or the closed state. It always opens to the same conductance state. The actual opening is stochastic, so we cannot predict exactly when a channel is open or closed, but we can say that there is a certain probability that the channel will be open. So this was published in 1976. However, as you can also see on that recording, it's quite a noisy recording. It doesn't sort of look like a very clean, beautiful recording. And indeed, the technique was not fully developed at that point. And actually, for 4 years, essentially nothing more happened. Various people tried the technique, it didn't really work very well, and most people said, oh, okay, they've showed the principle, we can see the channels, but actually in practice, we can't do anything about it. But then, one day, in the summer of 1980, Erwin Neher, who persisted in trying out how to improve the method, made a very important, although it may seem to you a trivial discovery, but sometimes very practical things can seem very simple in the retrospect. So people who worked with classical electrophysiology and who were used to use intercellular sharp microelectrodes would use the same electrode again and again. You puncture one cell and you measure something, and then you puncture another cell. And they again approach this in the same way. So you try to patch one cell and then you patch another one. But Erwin, who was obviously very observant, noticed that when there was a success, it was always only when he used the pipette for the first time. So he realized the pipette has to be completely clean. It cannot be contaminated by products from the membrane. Secondly, he sort of experimented, tried various things, and he discovered that if you apply a bit of suction to the pipette, then suddenly you can get a dramatic increase in the seal resistance between glass pipette and cell surface. And that was the discovery of what is in red letters up there, the so-called gigaseal, the giga-ohm resistance. And that turned out to be the real event. At that point, you not only had a fantastic seal resistance electrically, you also had the most fantastic mechanical interaction, so that the glass pipette actually was bound to the membrane very, very firmly. You could extract the membrane. You could just pull the pipette away from the cell, and the membrane would follow. Now you could actually do experiment on a completely isolated bit of membrane where you control the composition of solutions on both sides of the membrane. Almost a dream of all electrophysiologists. So with this improved technique, it was published in October 1980. And the reason I've written the right place at the right time, because I happened to be in Göttingen a few weeks after this paper was published. Erwin Neher had kindly invited me to give a seminar in Göttingen, and for the first time, I had obviously knew about the 76 paper, but like many other people, I thought, okay, it looks a bit difficult, it doesn't look very clear-cut, probably it's not worthwhile doing at the moment. As soon as I saw what things actually happened in Göttingen, then I realized the time is now. So first, with this improved technique, you see now the beautiful recordings that Bert Sackmann obtained from the motor end plate. Now the recordings are totally clean and clear-cut. And by isolating the membrane in the outside-out configuration, he could demonstrate that only when acetylcholine, the transmitter molecule, binds to the channel, it is open. In fact, two molecules need to be bound to the channel in order for it to be open. So this turned out to be a fantastic opening, and within a very short time, every electrophysiological laboratory across the world were applying the patch clamp technique. I obviously thought that this would now be the time to check whether ion channels actually really existed in epithelial cells and whether they played an important role in epithelial cells. Ion channels was essentially the preserve of neuroscientists at that time, and still to a large extent is actually. It was obvious that in a nervous system, the fire action potential to get the muscle to contract very quickly, you can only do that by activation of ion channels. It was not nearly so obvious that epithelia would have ion channels, and particularly that regulation of ion channels would be important. So we were the first, actually. This is one of those moments, and that may be another general lesson, that if you suddenly see an opportunity, sometimes it is necessary to act fast. And so we did that, actually, and we were the first to publish recordings of single-channel currents in epithelial cells, in pancreatic cells, and you see an example there from one of our 1982 papers. And so we could show that, yes, there are ion channels that actually behave much the same way as in nerve cells and in muscle cells, and they are regulated. But here's an interesting comparison when you compared Bert Sackmann's record with the one from the work that Yosho Maruyama and I did together. In the case of the nicotinic receptor on the end plate, you can see that acetylcholine needs to be bound directly to the channel in order for it to be open. If you look at the experiment to the right, you can see that we actually applied acetylcholine not to the bit of membrane from which we were recording, but from membrane outside the isolated area. And this isolation, as I pointed out before, is absolute. There is not a single molecule that penetrates this seal. So we had shown actually that acetylcholine opens this channel not by direct interaction with the channel, but via something happening inside the cell. So it's a messenger-mediated channel, and that turned out to be very correct. We then discovered the most important channel in the initial part of the work, namely the calcium-activated high conductance potassium channel, which is another channel that is activated by calcium. And in a certain configuration, this channel totally dominates the electrical characteristics of the cell. So it's basically in a certain configuration with a certain ionic composition. It is the only channel that exists from an experimental point of view. And this means that you can compare single channel current recordings that you see to the left with the whole cell currents which you see inside the red box. And that means that by comparing these two, you can actually calculate the number of channels per cell. So we have a very simple formula you see there inside the box. Capital I equals N times small I times P. P is the open state probability. So if the open state probability is 1, which means that the channel is open all the time, then the total current divided by the single channel current is equal to the number of channels in that cell. And this is a surprisingly low number, somewhere between 50 and 100. So compared to the number of receptors, which would be tens of thousands of transporters, it's a very small number. And that is why the patch clamp technique works, actually, because, as I said before, you isolate a small membrane area, and you have the good luck, which you can see to the left here, that there's just one channel in that preparation, and you can see, follow the opening and closure of that channel. So this is actually molecular biology in real time. You see the fluctuations of the protein molecule, that is the ion channel. Some of the conformations are open states. Some of the other conformations are closed states. So you can actually follow the movement of a single molecule, how it vibrates in this particular way. So anyway, to not make this too long a story, having demonstrated eventually that the chloride channel exists on one side of the cell, the potassium channel on the other side, we were able finally to produce a coherent model for fluid secretion in exocrine glands that actually applied not only to salivary glands, but to the pancreas, to the lacrimal gland, to the sweat glands. And the bottom line, and I don't want to go in detail with the model because it's probably not very important for most of you, but the bottom line was that the regulation of these two channels, calcium-activated chloride channels and calcium-activated potassium channels, allowed the active transport processes to proceed. So there was an active transport, but it was not a specific active chloride pump. Now, in very, very simple experiments on the isolated perfused pancreas, which you see in the middle at the bottom there, we could look at the requirement for calcium in the extracellular solution for secretion. And you can see here that there are actually two principal stimulants of acinar secretion, namely acetylcholine, which I already mentioned, and a hormone called cholecystokinin, abbreviated CCK. And here in this particular experiment, we use CCK. So we can activate fluid and enzyme secretion, but you can see that if you remove calcium at the point when we write calcium-free plus a calcium chelator, EGTA, secretion drops just like that. So it means that there is a requirement for external calcium in order to sustain secretion, and most likely this indicates that there is a calcium entry process which is important for keeping the calcium concentration high in order to keep the ion channels open. There's also another fluid secretion process which I will come back to at the very end of my talk. That is the duct cells. This is a cell that Peter Hecke is a world champion in looking at that cell, and it produces a bicarbonate solution. I will come back to that at the very end of my talk, and as you can see, this is not a calcium-dependent process. It still works in the absence of external calcium. We were also later able to record the actual calcium signals. You see that within the red box. And the calcium signals turned out to be short-lasting, repetitive calcium spikes rather than a sustained elevation. Now, then, of course, you start thinking about how could calcium get into cells. So you look for models elsewhere. And here's an example from the heart. Another example of excellence in Szeged comes up here, namely the beautiful work by one of our chairs, Professor Andras Varro. And in this review that I have quoted here, the left, published in Physiological Reviews 21, I recommend very warmly to everybody who's interested in physiology to read that review, because this is the most remarkable review of cardiac electrophysiology. And if you do not know cardiac electrophysiology, you will not know anything about how the heart functions. So that review is a must for everyone who wants to read it. It is actually also a fantastic example of real pedagogical writing. You can read this review and you can actually understand how the heart works. A beautifully illustrated, a wonderful example of excellence from the University of Szeged. Now, what I want to draw attention to here is the very bottom line. I put this red asterisk there. That is the way calcium gets into the cardiac cells. In the heart, we have very special type of action potentials, which you see at the very top left in this picture here. This is, by the way, also, this is human physiology, which is also very important. So you see this action potential, and you see this dramatic change in potential that occurs during the action potential in the heart, from minus 87 millivolts to plus 30 millivolts. And that opens up a voltage-gated calcium channel. So by this enormous change in electrical potential, you actually open the channel. So it's a voltage-operated channel. And then you get this calcium current, which is recorded here in the lower left part of the diagram. You can also record the single channel currents. It is turned to the very left from another good friend of mine, Harald Reuter from Bern, who sadly died some years ago and who made these beautiful recordings. Now, it was immediately clear when we consider this is the most famous calcium entry channel, that this would never work in the exocrine glands because we do not have this gigantic potential change. So even if the channel existed in the pancreas or in the salivary gland, it would never become activated. So things work differently there. And this is just a short illustration of how different these cells are in this particular respect. So here we record the calcium signals electrophysiologically by monitoring the calcium-activated chloride current. And you can see that the calcium signals are basically, as I already mentioned before, repetitive, short-lasting spikes. And what is important in this experiment is the first part of that experiment is carried out in the absence of external calcium. And you see it works perfectly well. So it means that a calcium signal in the exocrine epithelial cells is primarily generated by release of calcium from intracellular stores. And this is illustrated in the red box to the right. And it was actually in the pancreatic acinar cells that Mark Barrett and his colleagues discovered the messenger that releases calcium from the cells, namely this small molecule IP3 inositol 1,4,5 trisphosphate, a small water-soluble molecule that is generated by agonist interaction with certain receptors in the membrane. And actually another important discovery by a Hungarian scientist, András Speth, in Budapest at the Semmelweis University, who was the first to demonstrate the IP3 receptor in the endoplasmic reticulum membrane. So we had to look for a different entry mechanism, and it turns out that the way calcium is gated in these cells is as a consequence of the release of intracellular calcium. And this is illustrated diagrammatically. It's a complicated process, but it has its merit. So acetylcholine interaction with the receptor generates IP3. It binds to the IP3 receptor, and the receptor opens. It is a channel that is permeable to calcium. Calcium is released from the endoplasmic reticulum inside the cell, and by lowering the calcium concentration inside the endoplasmic reticulum, there's a conformational change, and a calcium sensor in the endoplasmic reticulum membrane, green STIM1, will bind directly to a completely different calcium channel, also known as RI1 and known as a crack channel, where crack stands for calcium release activated calcium channel. And we managed for the first time, actually not that many years ago, a bit more than 10 years ago, to finally be able to record that current in the pancreatic acinar cells. It's a very, very tiny current, and you cannot get single-channel recordings. It's simply not possible to get that resolution. But we could record this particular current. And we also found various small molecules that can inhibit that current, which is also a little bit important. Now it turns out that the normal physiological calcium signals that will generate secretion, as I mentioned before, is not a sustained elevation of the intercellular calcium concentration, but these short-lasting spikes. However, if you generate a sustained elevated calcium concentration, you will kill the cell. So this is an example of a tightrope balance that all cells have to work. They need to use calcium as a signaling molecule, but if there's too much of it, it actually is dangerous. And one of the ways you can create these big signals is by something related to alcohol. And we have basically two different inflammatory conditions in the pancreas that are important in human disease. And we have alcohol-related pancreatitis, and we have pancreatitis related to gallstone complications. I will only talk very briefly about the alcohol-related pancreatitis. I thought initially, and somewhat naively as it turned out, that if I just put isolated pancreatic cells in a concentration of high alcohol, then they would destroy. But actually nothing happens, mostly when that happens. However, if you combine alcohol with long-chain fatty acids and generate fatty acid ethyl esters, as illustrated there, then you generate a very powerful calcium releaser. And then you get these big sustained calcium signals. If they are sustained for half a minute or a minute or something like that, then the cell will die, and will die in necrosis. It will explode. It will be an inflammatory event. So the solution to that would then be to put a brake on calcium entry, and indeed then we applied our knowledge that there is this crack channel or I1 channel, and if you could inhibit calcium entry through that channel, that might help. And indeed it does. It reduces the calcium elevation to a more physiological level. It markedly reduces the protease activation inside the cell that begins to digest the cell itself. And importantly, as you see in the lower left part of it, in a concentration-dependent manner, it dramatically reduces the level of cell death and necrosis. So this is a kind of proof in principle. Now there are clinical trials going on to see whether this would actually be the way of treating acute pancreatitis, which up to now is, relatively speaking, untreatable disease. Nevertheless, many people die of it many years, and it is a precursor to pancreatic cancer, which probably is the most unpleasant way to die. Now I want in the last minute to shift gears a bit, but we will actually see in a moment that it comes back again to some of the same problems. Before I do that, just to say that this inhibition of crack channels works not only because you can inhibit calcium entry into the acinar cells, but also because you can interfere with calcium entry in neighboring cells, in stellate cells, in inflammatory cells. And these cells also play a role, particularly the inflammatory cells that enter the pancreas in the initial state of acute pancreatitis play a big role. So this crack channel inhibition in these three cell types is probably quite important in the disease process. But now to another disease, which is also an exocrine gland disease, namely cystic fibrosis. This is one of the very few examples that we have of a very simple monogenetic disease. So there's one gene that is affected, and if you have two copies of that defective gene, then you get disease, cystic fibrosis. Now we go back in time, a long time, to another friend of mine, Irene Schulz, born in Berlin, working in Germany most of her life, but having a short sabbatical at the NIH. And you see a diagram of the sweat gland down there. So, like all the exocrine glands, the sweat formation is a two-stage process. You produce a primary fluid, which is basically sodium chloride solution. And then in the duct system, in the sweat duct, you reabsorb salt without water. So the membrane is relatively water impermeable, and you basically extract salt from the solution. So in the normal sweat, the salt concentration is relatively low. And this is advantageous. We use, of course, sweat formation to get rid of heat because the water will evaporate, but we minimize the loss of salt. Now, it has been known since the Middle Ages that when you kiss a baby and it tastes salty, it's a very bad sign. So there's a lot of medieval poetry in Germany, in Spain, and many other countries that testifies that observant mothers knew that when they kissed a baby and it tasted very salty, that baby would die quite soon. And that is indeed the case, because what Irene Schulz discovered in '69 was that whereas the primary secretion, which she obtained by micropuncture, was completely normal in cystic fibrosis, the final sweat was absolutely, totally abnormal. In fact, it had a very high salt concentration. So she reasoned that because the electrical potential difference across a duct was not diminished, but actually enhanced, she reasoned that probably there was an active sodium reabsorption, and chloride should have followed passively if the chloride permeability was high, but that in CF, the chloride permeability might be low, and chloride could not follow. So you have the big potential. It tries to drive chloride across the epithelium, but it cannot work because the permeability of a chloride is lacking. And that turned out to be right. This was actually a premature discovery that what is wrong in cystic fibrosis is that the chloride channel is deficient. This was forgotten completely by most investigators in cystic fibrosis, and the whole field was for many years in complete disarray because people did not understand what was happening. And then suddenly, molecular biology brought a great deal of excitement to the field when John Reardon and his collaborators in Toronto discovered the genetic defect. And they found out that the vast majority of people who suffer from cystic fibrosis have a defective protein, as written there in the abstract from the science paper, that at position 508, there is a phenylalanine amino acid that is missing. However, that does not by itself tell you what that molecule does. So because it happened that way, this gene that encodes the chloride channel, which is a completely different chloride channel from the chloride channel I talked about. It's not a calcium-activated chloride channel. It is a cyclic AMP-activated chloride channel. But that normal gene product got the bizarre name cystic fibrosis transmembrane conductance regulator. And this is a crazy name, which is abbreviated CFTR. So we have the absurd situation that a normal gene is named as a disease-related entity, which, strictly speaking, is complete nonsense. But the name stuck, and so we are still stuck with the CFTR. In any case, John Reardon cleverly realized, even before they finally had this result, that maybe this channel was a chloride channel. And so John Reardon calls me one day and says, there is a girl in my laboratory, her name was Christine Baer, and I would like her to do some electrophysiology on that molecule to try and find out what it does. Okay, I say it's fine, send her to me. And so she did come and stayed in Liverpool. I was working at the time for about a year. And we did, I showed, she had never done electrophysiology before. So we taught her electrophysiology and we did some work together and published it. There was nothing very sensational about it. But she learned how to do single channel recording, went back to Toronto, and a few years later she caused a major sensation by publishing this paper in Cell in '92 that directly proved that CFTR is a chloride channel and is a cyclic AMP activated chloride channel. And that was a decisive breakthrough in CF, because finally we knew what the molecule that would be wrong in the disease actually was doing. And here you see her recordings in the lower right part. But then came the surprise. The initial expectation was that in CF there would be no channel at all. And that turned out not to be the case. As you can see to the right, this is from a defective CFTR with a delta F508 mutation. The channel does exist there. It does open, but it does not open very often, and it doesn't open for a very long time. So the open state probability of the channel in the disease state is much reduced. And it turned out that there are basically two problems in CF. On the one hand, the defective protein is not processed in the normal way, so much less of the channel is inserted into the membrane. And even when it is inserted in the membrane, it does not open nearly as often as it should do. And by a search for molecules that could modify that, a number of small molecule modulators were found that could actually correct both these defects. So we have drugs that force the molecule to be inserted in the membrane, and we have other small molecules that markedly enhance the performance of the channel when it actually gets into the membrane. And this is now effective treatment in cystic fibrosis, which has transformed really the fate of patients who usually died very early on and now can live to a much riper age. And this finally brings me back to my final slide here, to the pancreas again. Because in the pancreatic duct cells that produce the bicarbonate-rich fluid secretion, the most important chloride channel is not the calcium-activated chloride channel that I talked about in the Eisner cells, but CFTR, as you see in the upper left part of the diagram. That is the dominant chloride channel, and that chloride channel is essential for the ductal fluid secretion in cooperation with the chloride bicarbonate exchanger. And more recently, Peter Hegge and his group made a very important breakthrough in the understanding of alcohol-related pancreatitis by demonstrating that high alcohol levels, and also often in combination with the fatty acid, as I mentioned before, will markedly inhibit the function of that channel. And that's very important actually, because we need these enzymes that are produced and secreted from the Eisner cells to be washed out of the system as quickly as possible so that they cannot begin to digest the pancreas itself. These enzymes should digest the food we are eating in the intestine. These enzymes should not be used to digest the pancreas itself, which is what happens in acute pancreatitis. So that paper published in Gastroenterology in 2015 is a very important paper by demonstrating yet another action of alcohol. And when we started to work on the human pancreas in the lower left part of the diagram, we discovered that if you got a pancreas from surgeons where there had been obstruction of the pancreatic duct, then there was enormous amount of trypsin activity in the Eisner cells. So it illustrates really the importance of having the ductile flow of fluid and bicarbonate, which Peter and Sultan and the colleagues in Szeged had done so much to show. Another example of excellence in Szeged that has contributed to the reputation of this great university. So finally, on the left side, the people who did the work in my laboratory, and to the right, the many people who inspired me and continue to inspire me. And once more, so many thanks to Peter particularly for many years of friendship and collaboration in some ways, but I've always enjoyed our talks and our discussions, which have also helped a great deal to clarify our minds. So thank you for your attention.
Thank you very much. Thank you very much, Ole, for this fantastic talk, and I think it's a really very good showcase what you have shown, that first we need to understand how systems works, and maybe just the understanding or discovery when we can use this knowledge comes much later, because plenty of times just the understanding is the key factor. So now it's time for questions. I would like to ask the students whether who has any question.
So you mentioned that we cannot know, if I understand it right, we cannot know whether a transport channel is open or not. Does that have anything to do with probability theory, so that the fact that we cannot know the state of an electron, or is it a completely different mechanism?
Sorry, it's very, very difficult. I don't know if anybody can help, if you could repeat it, because I just couldn't.
So you said that we cannot know exactly whether an ion transport channel is open or not, right?
So he is asking whether we cannot know whether the channel is open or not.
I mean, you mean whether how we know whether the channel is open or not? Well, I mean, as I said, the channel opening, and this is universal for all ion channels, it is a stochastic process. So we cannot, let's say, make a statement that in 20 seconds this channel will open. Nobody, however experienced he or she is in single channel recording, can make such a statement. We don't know. It's a stochastic process. But with accumulating a lot of data, we can say under certain conditions that there is a certain open state probability. It can be 70% of the time that the channel is open. With an increased calcium concentration, the channel may be open 95% of the time. So that's why we talk about this small p-value. The open state probability is the way we measure the channel activity. But when we make wholesale recordings, of course we are recording from a composite of ion channels, and then we can make confident predictions. If we look at a single channel molecule, then, as I say, it's a stochastic process. It reverberates in ways which we still cannot fully understand, but we can gain insight into the overall regulation. So basically, it's a regulation of a on rate, the rate at which the channel opens, and there is an off rate, the rate at which the channel closes, and the mixture of the on rate and the off rate will determine what the open state probability is. I could not hear you correctly, so I don't know whether my answer is an answer to your question, but anyway, that's what I can say about the opening of ion channels. Ion channels come in many different shapes. I mean, as I said, it can be ligand operated, they can be voltage gated, they can be messenger mediated, and in some cells, like in Andras Varro's heart, there are very many ion channels present at the same time, and there's a rich opportunity for pharmacological intervention.
I see. And could it have anything to do with the probability theory of electrons?
So he's asking that whether the probability of the electrons movement could act on the channel.
You mean whether the movement of ions by itself could change things? Yes, I mean, that certainly is the case. By changing ionic composition, you can get a channel to behave very differently. If you remove all the calcium from the solution, for example, a calcium channel will actually conduct sodium. So yes, the individual ionic composition can certainly change the characters of a channel.
Okay, thank you.
Thank you. Any other questions from the audience, students?
Yes, there's one there.
Yeah.
I can see. Long distance.
Thank you very much for your presentation, Professor. My question would be, is there any other way that we could drain fluid from the pancreal duct beside the CFTR to prevent fluid buildup and prevent maybe pancreatitis, acute pancreatitis?
I didn't hear.
The question was whether any other way to wash out the enzymes from the pancreas than the CFTR provides fluid outflow.
I see.
To prevent pancreatitis.
I see, yeah. Well, of course, I mean, there are two separate fluid secretions, as I hope you understand, from the Eisner cells and from the ducts. Both of them are important. The ductal fluid secretion is probably particularly important in terms of this, so this is important. But of course, you can prevent the activation of the enzymes inside the Eisner cells. And that's why we think that the inhibition of the calcium entry channels is a way of lowering the calcium concentration inside the cells, and that will potentially prevent the activation of the enzymes. And we know that because we can measure the activity of the enzymes inside the Eisner cells. So we know that if the calcium signals consist of interrupted spikes, there is no activation of the enzymes inside the cell. But if the calcium concentration is kept high, constantly, then you actually get, and we can look at that in real time because we have fluorescent probes that will show us whether the enzymes are active in the cell. So that's why these potential inhibitors of calcium entry channels may be a solution, and they are being tested, and Peter group has also worked on that. It may not be the only solution, but it may be part of the solution.
Thank you very much.
Thank you very much. I also have a question because many students are here also, or even supervisors, mentors, and I found two very interesting things in your CV. Actually, one of them, when the patch clamp new technique came out, even you couldn't buy the equipment. You had to go to understand what is it equipment, and almost everybody believed in that this will work in, salivary glands and it has any effect on the secretory. So it was extremely risky, and what, did you have any fear, during this period? Everybody said it will not work. Maybe from 100 people, 99 said it will never work. And you believed in, and have you thought ever that maybe this can, lead to stop you in science if results not coming? And my other question is, you also changed institutions during your career. Did you have also any fear, when you go there, they expect you to be successful, you would like to go through, and if the data is not just not coming?
Yeah, so the first part of your question is very perceptive, as always. So when I gave this seminar in Göttingen in the early November 1980, and I was impressed by what Erwin and Bert showed me, then of course you have to make a decision. I mean, it's just an interesting visit and give you some kind of general impulses, or is it time now actually to, as you say, invest your energy in this and say, well, this is it. Yeah. So there's a risky moment. And interestingly, I came back to Liverpool full of enthusiasm. And the best person I had in my lab at that time was Yosho Maruyama, who had relatively recently arrived from Japan, but he had already started on another project. So I told him what I had seen in Göttingen, and I said, well, the time is now, we are going to do this. And he was not very happy. No, he said, well, exactly as you said, how are we going to do this? We have to construct an amplifier. We cannot buy the amplifier. I have never done any electronics in my life. He was a medical graduate. So for him, this was a huge challenge. Well, I said, we have the drawings from Erwin about how this can be done, and you will do it. I was very busy at the time because at the same time I was moving from Dundee to Liverpool, so it was very much up to him to get the thing started. And he was very keen, very understandably, to finish the other project that he was doing. And he saw exactly, as you say, well, I'm a short-term visitor here. I will end up in a project that doesn't work, and I don't have a single paper or anything like that. Anyway, maybe happily, because he was Japanese, and the Japanese have a certain respect for authority, and so finally he had to agree that, yes, that is what we are actually going to do. And again, and that's a very important point that my good friend Ole Petersen in Kiev often has made. We actually often have abilities that we don't know we have. And it turned out that Shumayama, a straightforward medical graduate, actually when he came to me, he had never done an experiment before in his life. He just expected to be a medical doctor, but somebody thought he should go and get some experience. And, well, he took these diagrams. He went to the library. He got electronic textbooks out. He went to the electronic engineering department and asked people, and he produced our first amplifier. He blew quite a few operational amplifiers in the process, but they were not very expensive. And so, in a relatively short period of time, he actually had constructed the amplifier that we needed. And he also learned how to isolate cells. We've never done isolated cells before. And so now I often meet students who come to the lab and say, somebody has to show me how to do it. I don't know how to do it. And I said, well, you know, there was somebody who tried it for the first time. And they did not have anybody to show you how to do it. And that's one of the dangers of always thinking that you have to have a course. Somebody has to show you how to do things. So it was risky. And yes, I took the risk in a sense, but it was also a risk for him. But it turned out very greatly to his advantage. He had got 5 papers in Nature, and he came back to Japan as a great hero, as it were, and became chairman of his department in due course. So yes, there's no gain with it, or a certain risk. In terms of moving, yeah, that's the same again. You think, is it a good thing, is it a bad thing? And you actually, in that case, don't really know whether it is a good thing because you don't have the control experiment. Maybe if I stayed in Dundee rather than going to Liverpool, it would have been equally good. But you do what you feel like in a sense. I think in many of these kind of decisions, whether you move to one place or the other one, in retrospect, it will probably often turn out to have been quite unimportant. And you might have done as well if you had made a decision in another way. But a decision about what you are doing next as an experiment, and we talked with Martin about that earlier at lunchtime, that is the important decision. And nothing is more important in science than deciding what do you do next. And there's a risk there.